In this lecture, we're going to talk about HIV, which stands for Human Immunodeficiency Virus. And what makes HIV so deadly for the human immune system is the type of lymphocyte that it attacks. HIV specifically attacks a helper T cell. So in this illustration here, this is HIV. And this guy here is a helper T cell. And on the surface of a helper T cell, you have a receptor. And this receptor is called a CD4 receptor. This is the receptor that a surface molecule on an HIV virus, a surface protein on an HIV virus, specifically binds to. So HIV will bind to the CD4 receptor. And within HIV, you have these like little squiggly lines right here. This is viral RNA. HIV is what we call a retrovirus. And it's a retrovirus because it uses RNA as its genetic material as opposed to using DNA. Also inside this HIV virus, this little yellow dot right here. That yellow dot is called reverse transcriptase. So you'll have to kind of remember back in your biology days about a process called transcription and translation, which was using DNA, the blueprint, to produce proteins. Okay? So reverse transcriptase is going to have a role in that process. Okay. So once this HIV virus binds to a CD4 receptor on a helper T cell, the membranes of the virus and the membranes of the helper T cell fuse together. And by doing this, the HIV viral RNA and the reverse transcriptase are released into the cell. So here you have viral RNA and reverse transcriptase being released into the cell. And now what happens is that reverse transcriptase, that reverse transcriptase converts viral RNA to viral DNA. In other words, reverse transcriptase transcribes viral RNA into a viral DNA. And then that viral DNA will migrate itself into the nucleus of a helper T cell. So here you have viral DNA. So this is viral DNA. And that viral DNA will fuse itself into the host cell's DNA, right? And then what happens is transcriptase. So transcription. Probably can't read that. Let's change that up a little bit. Okay. So we have transcription take place. Transcription. Okay. So transcription of this new DNA molecule that includes the viral DNA molecule will produce this thing right here. Right? That new thing is what we call viral mRNA. Viral mRNA. And that viral mRNA will be sent to this guy right here. This is a ribosome. A ribosome is an organelle that makes proteins. So this viral mRNA is delivered to this ribosome, and this viral mRNA codes for the message to make these guys here called viral proteins. And those viral proteins will be assembled. And they'll be assembled into a new HIV virus. And when a new HIV virus or the insides of an HIV virus are created, then it buds off, ready to infect another helper T cell. So this here is a new HIV that buds